Hello again. So now we continue with Parkinson's disease. So, Parkinson's disease, and again, I'm going to assume that you remember what Ahmed has said about Parkinson's disease, so I'm not going to go into any details about, the disorder itself, but mostly about the genetics and the molecular biology, what's happening to the cells, to the proteins, etc. So, Parkinson's disease is thought to have some genetic component. But you see, it says some genetic component. Alzheimer's disease, even the sporadic versions, is thought to be sort of 70%, whereas Parkinson's disease is actually less genetic than Alzheimer's. So the heritability is only 30%. And again, the majority is the late onset sporadic version. So that's 85 to 90%. So in one way, Parkinson's disease actually does have sort of 10 to 15% of patients having mutations in particular genes that cause the disorder. So with Alzheimer's, it was mutations in APP, PSEN1, and PSEN2. And that only caused about 1% of Alzheimer's disease in patients. However, for Parkinson's disease, there are actually more genes that can be mutated to cause the disease, and it causes more of the disease. So 1% in Alzheimer's versus 10 to 15% in Parkinson's disease. But overall, particularly in the late onset sporadic cases, the heritability is only about 30%, whereas in Alzheimer's it was 70%. So it's these risk factors. There's more genetic ones in Alzheimer's where there seem to be more environmental causes maybe in Parkinson's. And just to say, well, actually, I'll go on about that a bit more in the next slides. So there are some genes and loci associated with PD risk, but it's not nearly as many as with AD. And again, the genetics give more information about what might actually be happening in the brains of the patients. So, we know the pathophysiology, like we know the dopaminergic neurons are dying, there are Lewy bodies, they're dying in certain places. That's what causes the effects on the movement of the patients, the substantia nigra involvement. All of that is known from looking at the patient brains. But by looking at the genetics, you can maybe start again to figure out, well, why do these things happen? And hopefully with a better understanding of why these things happen, you can design better treatments for patients. And so again here, the genetics give more information and add to what's known about the disease and can give ideas about the pathophysiology. So this is what I was going to say in the last slide, and I thought, no, I have a slide on that, I'll wait for that. So Parkinson's disease is partially genetics. There are 26 is kind of high. There are possibly 26 individual genes that can be mutated to cause Parkinson's disease. Not all of them have been validated, so it's a bit lower than that. And then as far as the environment, what we mean is not like, is it raining or not, but things like pesticides, head injury. Does the person smoke? Does the person drink coffee? Does the person drink alcohol? I mean, they're just various, what about chemicals that are, in the environment, that are in cleaners and things that we use, things like that. That's what the environment means. It doesn't mean like you live in a hot climate or a cold climate. I think environment is a term that's used and people don't necessarily understand what it means with regards to disease. And then there's also epigenetics that's thought to have some effect. So DNA modifications, regulatory RNAs, histone modification, that sort of things. You may, if you were here last semester, you may have gotten some of that in the genomics and bioinformatics module. So you'll kind of have a good idea of what that is. And so the idea is that it's, some of these risk variants, genetics, some of the environment, and some of the epigenetics all together, that's what causes Parkinson's disease. And so it's really complicated, and like AD and a lot of neurodegenerative diseases, it's really unclear a lot of the time how exactly, it's caused and what exactly it is that causes the neurons to die. And I should also add here, because I didn't talk about this with AD, and it applies there as well as with PD, the main risk factor overall is age. So, people who are older are more likely to get neurodegenerative disease, AD, PD, and possibly some of the other ones as well. So, what do we know about Parkinson's disease? So this is some of what you should have gotten from Ahmed already, so I'm not going to go through it in too much detail. But alpha-synuclein is something that is important in Parkinson's disease. Generally, the protein is highly expressed in neurons. In the disease, it appears to misfold and form aggregates that become Lewy bodies. Lewy bodies are a key indicator of PD in brain tissue, though not all PD has Lewy bodies, though most of it does. The normal function of alpha-synuclein is not really known, though some of the research has shown that it may be responsible for presynaptic maintenance of the synaptic vesicle reserve pool, so taking care of the synapses before they get sent into the vesicles, the neurotransmitter vesicles that will get put into the synapse. So alpha-synuclein's not the only thing in the Lewy bodies. There's ubiquitin, heat shock protein, HSP70, but how any of these are involved in the pathology is unclear. Why it's the nigral dopaminergic neurons that are mostly affected also is unclear. And there are a lot of theories, again, for Parkinson's disease, and I'll go through some of them, and many of them are based on mitochondrial dysfunction and oxidative metabolism. So for Parkinson's disease, because it has less of a genetic basis, it was only recognized as having any kind of a genetic component at all in the late 90s. So until the SNCA mutations were found in patient families that caused the disease, people did not think Parkinson's was genetic at all. So unlike Alzheimer's, which people thought was genetic from very early in 1930s, I think I said, Parkinson's, they didn't really think it was genetic at all until very much recently in the late 90s. I mean, more recently to me than you, I think, but there you go. So the first gene that was found, and I'll go through these in more detail in the upcoming slides, was SNCA. The second one was parkin, and then one a bit further along was PINK1. Parkin and PINK1 particularly support the mitochondrial hypothesis. SNCA is actually the gene for alpha-synuclein, so that sort of added and made sense with the theories of Lewy bodies and the fact that Lewy bodies are found. And it was only sort of the mid to late, I guess, 2000s that the GWAS started, so almost 2010s. And here you can see that different GWAS studies have ended up coming up with a lot of different genes that might be involved, and these ones that are here are some of the ones. Not sure all of them have been fully validated. That might actually be the difference between the boxes and the ovals. But yeah, there are on the order of about 20 or so individual genes that cause familial PD that have been identified. And yeah, sorry, the genome-wide association studies are looking for these genetic risk factors. So this is kind of an equivalent graph as to what I showed before, where across the x-axis we have the rare versus the common, and along the y-axis we have low, intermediate, or high risk, essentially. They call it penetrance here, but it's actually kind of risk of having the disease. So SNCA, VPS35, LRRK2, these three are autosomal dominant disorders. DJ1, PINK1, and PARKin are autosomal recessive disorders, and so that's what the difference in how they're pictured are. The ones with the half and half are ones where one mutant copy causes the risk or the disease, whereas here both copies need to be mutant to cause the disease. And as you can see, there are more genes in this category than there were for AD. So if you remember, for AD, it was just APP, presenilin 1 and presenilin 2. With PD, there are a lot more genes that, when they're mutant, can cause PD. But again, these are very rare. So if you remember, it was about 5 to 10% of total cases. And that's across all the mutations in all the genes. So there are some alleles that then cause intermediate risk. So they're high relevance sort of risk factors. And then there are the ones that are identified in the GWAS studies, which are common risk variants. So now I'm going to go through some of these genes in a bit more detail. I think SNCA, PINK1, PARKin, DJ1, and LRK2, just to tell you a bit more about the genes and what they do and how they might seem to cause Parkinson's disease, though it's really not necessarily clear all along. So the SNCA mutation, which is also sometimes identified as PARK1, these PARK indicators are kind of the order the genes were discovered in. It's, as I said, autosomal dominant PD, and that means one copy of the mutation is, one of the copies of the gene, so we all have two copies, one of them has to be affected. So people with mutations affecting SNCA, alpha-synuclein, have early onset Parkinson's disease, and they have a good early levodopa response. The hallmark of this disorder is, of course, Lewy bodies. SNCA, the gene, is found on chromosome 4, and like I said, it encodes alpha-synuclein. And as you might expect, or, if you think about it, it makes sense that rather than mutation that would decrease alpha-synuclein causing the disease, it's actually duplications and triplications of the gene that cause the disease. And so, because, if it's, the aggregates that cause the disease, if you have more alpha-synuclein present, you might be more likely to form the aggregates and more likely to cause the disease. And so I thought this was a fairly nice indicator of what alpha-synuclein might be doing in healthy neurons, alpha-synuclein is thought to maybe be involved in autophagic lysosomal clearance, oxidative balance, ER function, mitochondrial function, and synaptic function. And so these little yellow things are the alpha-synuclein protein. And then in Parkinson's disease, you get these alpha-synuclein aggregates, and what happens is synaptic dysfunction, mitochondrial dysfunction, oxidative stress, aggregation of the proteins, autophagic lysosomal disruption. So these proteins, these aggregates, aren't properly degraded, and also ER dysfunction. So PINK1 is actually also known as PARK6, and it is one of the autosomal recessive Parkinson's disease. I'm not going in order of them being found, and you'll see why in a minute. PINK1 stands for PTEN, induced putative kinase 1, and you can also, expand the PTEN, but it doesn't really give you much more information, so I'll leave it at that. It is a serine threonine protein kinase, so it's one of these proteins that adds phosphate groups to other things on their serine threonine amino acids. And most mutations, unlike with alpha-synuclein, where the mutations cause an increase in amount of alpha-synuclein, PINK1 mutations actually affect the kinase domain, so they mean they are loss of function mutations. They cause the gene, the protein, not to work anymore. And so that's not surprising because it's autosomal recessive. That means both copies of the protein, both copies of the gene need to be mutant before the effect is seen in patients. So this is just a diagram of what happens. So it's thought that PINK1 will go into the mitochondrial membrane, essentially. It'll get cleaved by a protease, and that will cause it to get degraded by the proteasome, ubiquinated, and then degraded by the proteasome, which is a protein degradation pathway. When the mitochondria themselves are damaged, it's thought that this import doesn't happen, and so the cleavage doesn't happen, and then the PINK1 actually accumulates on the outside of the mitochondrial membrane, and this recruits parkin. Parkin is important because that's another one of the genes that, when mutated, can cause Parkinson's disease. So we'll look at that a bit more. And that's why I've talked about PINK1 first rather than the order they were found in. So next we have parkin. And so it's known as PRKN, parkin, and PARK2. So this was the second one that was identified. And it's actually the most common cause of ARJP, which is autosomal recessive juvenile Parkinson's, and the most common cause of generally early onset Parkinson's disease. So again, mutations in both copies of the gene that make it stop working really has an effect on people, and it causes Parkinson's disease to arise much earlier. So what is parkin, the protein? Is it a kinase? No, it's an E3 ubiquitin ligase. What is an E3 ubiquitin ligase? It's a protein that adds ubiquitin molecules onto proteins to tag them for degradation. And that's part of the proteasomal pathway normally. Proteins get ubiquitinated by E3 ubiquitin ligases. Parkin's not the only one, there could be other ones, and then that sends them to the proteasomal degradation pathway. So, what we think might be going on with parkin and PINK1 in patients is that We think that PINK1 and parkin may work together to cause mitophagy, which is autophagy of mitochondria. It's a new word, mitophagy. So essentially, PINK1, parkin work together to get whole damaged mitochondria degraded. So how is this thought to work? We've got normal mitochondria, which are green, damaged mitochondrion, single damaged mitochondrion, which is yellow. How is it damaged? It might be any of these things: mitochondrial DNA mutations, increase in reactive oxygen species, decrease in mitochondrial membrane potential, misfolded proteins. All of these things, they're not really sure, could indicate that this is a damaged mitochondrion. And then, like I said in the previous slide, PINK1 accumulates on the outer membrane. PINK1, if you remember, is a serine threonine kinase. So it phosphorylates ubiquitin, it phosphorylates parkin, it phosphorylates, so the ubiquitin chains that are actually on the mitochondria. Parkin, when phosphorylated by PINK1, we think that recruits the parkin also to the mitochondria. And then the mitochondria has these phosphoubiquitin chains, and other proteins are also ubiquitinated by parkin, and that tags this mitochondrion to be identified and located by the autophagisome, which then causes what we're calling mitophagy, joining with the lysosome and degrading the entire mitochondrion. So that is one of the theories. Again, Parkinson's disease is like Alzheimer's disease, and there are a lot of theories surrounding how the neurons might actually be dying, why they're dying. And this is one of the theories. It's not absolutely certain that this is how it works. So this figure is from I can't remember the year, but it was 2017 paper then. So so it's still being worked on, but some of this stuff has been worked out in mammalian cells, and some of it was worked out in Drosophila, actually, which I think is cool, because I work on Drosophila as a model system. Actually, this is the work that got me into using Drosophila as a model system, working on Parkinson's disease, because I thought this was fascinating. But exactly what's happening in patients, we're not really sure that this is what's going on, but it could be. So to look at it in a bit more detail, we've got PINK1 that's been stabilized at the membrane of the damaged mitochondria. It phosphorylates parkin, it phosphorylates ubiquitin, so parkin gets attached to the mitochondria, adds ubiquitin to various substrates on the surface of the mitochondria, and that causes the mitochondrion to go through mitophagy. Like I said, there are other theories of how PINK1 and parkin might be working. Another one of these theories is around Paris, and I don't know what that stands for. You'll have to look it up, but here's a reference. Essentially, parkin doesn't just ubiquinate proteins on the surface of mitochondria. So, obviously it's in the cell. It might be targeting other proteins, and it's thought that parkin ubiquitinates a protein called Paris. It could be that, it is thought that PINK1 phosphorylates Paris, and then that's what causes parkin to ubiquinate Paris. Paris then gets degraded by the proteasomal system, and that offers neuroprotection. And that's what happens is thought to happen in the normal case. And so the way that Paris became of interest is that it was found that in patients with Parkinson's disease, Paris is another protein that accumulates. So it was found that Paris accumulation is seen in sporadic patients with Parkinson's disease. And so, so then it was identified as a target of parkin, and then it's like, okay, well, maybe that's what's causing the disease. And so in parkin and PINK1 mutant families, you can say, oh yeah, that's why Paris is accumulating. But it turns out that in people with sporadic Parkinson's disease, with Parkinson's disease that isn't caused specifically by mutations in genes, these proteins can also still get inactivated. So it could be caused by mutations, like I said, but actually oxidative stress and other cellular stressors can inactivate these proteins. They seem to be susceptible. And so Paris will accumulate, and then Paris actually goes into the nucleus and affects the transcription of PGC1 alpha. And so if it accumulates and goes and binds the gene, it'll actually stop it from being expressed. PGC1 alpha is important for creating new mitochondria, and maybe the cells don't have enough mitochondria, and that's what causes the neurodegeneration. So again, another also mitochondrial-associated pathway. That's a theory of what might be happening in patients with Parkinson's disease. All right, I'm going to stop there, and then we'll be back in the next section to just talk about the last part of the Parkinson's disease lecture. I'll see you when you start the next video. Thank you.